Tom was a project manager and research associate under the Canadian Glycomics, if I pronounce that right, and network and Semnana Cystomics. Both sound very interesting. So without further ado, let me welcome Thomas to give us a seminar today. We look forward to learn from you about your research work.
All right. Yeah, thank you so much, Wei. I appreciate the lovely introduction. Why don't I start out by sharing my screen? Looks like I should be able to do that. Perfect. Okay. So yeah, as Wei was explaining, I'm really interested in bacteriophage-based biocontrol of Salmonella, and I'm going to be talking about that sort of through the lens of a One Health approach through the talk today. And then you might see throughout the presentation as well some QR codes. You can see one on this slide. So if you would like to scan at any point, this one, for example, would connect you to my LinkedIn. If you have any questions, you can always reach me there. Or there might be some others where it links to papers that are relevant. So feel free to scan those as well. And then I have some other work that's coming out soon. So feel free to reach out if you are interested and you'd like to see where things are going as well. Okay, so just to give you sort of a brief overview of sort of a bird's eye view, if you will, of what we're going to be looking at. Just going to set the stage, the context of One Health, why it is that Salmonella in chicken is a problem worth addressing. It's certainly one we've all heard of. You know, you need to cook your chicken, and the reason is usually Salmonella. And then also why bacteriophages are could be a nice alternative antimicrobial to target Salmonella in chicken, and also why now, why not before. So hopefully I can cover all of that in sufficient detail today. So just right away, sort of to set the stage, the World Health Organization basically just says that One Health is an integrated, unifying approach to balance and optimize the health of people, animals, and the environment. And you can see in that nice little figure on the side there how they overlap and One Health is in the middle. So the One Health approach is particularly relevant, I would say, for food and water safety, nutrition, the control of zoonoses, so diseases that can spread between animals and humans, pollution management, and then combating antimicrobial resistance. So you can see all of these things already. They match sort of the presentation I'm giving today quite well. So basically focusing on the emergence of microbes resistant to antibiotic therapy is definitely a big one to think about. So our research really digs into that combating antimicrobial resistance, or you might hear me say AMR throughout the talk. And that's sort of the aspect of One Health that we're most concerned with. But it's important to state also with One Health, each of these domains is very large, and we can't do, everything all at once. AMR is a huge problem, so we have to sort of focus in and find that one project that might span some of these different borders, but that can be sort of effectively targeted and implemented. So it's about finding that single sort of identifiable problem and then focusing in on that domain through strategic collaboration as well, because there's information that these different groups might have. So for example, you can see on the one side, we have our environment, our human health, and our animal health, how they overlap. So what does this look like in the real world? Well, it would be maybe government monitoring programs. It could be producers and processors of different food products. It could be hospital data, and then basically watching for things that are traversing through each of these different domains and then finding out why, and then also coming up with a good strategy as to how you can actually target the issue that's spanning. So the use of antibiotics in animal agriculture is recognized as a major contributor to the development of AMR, or antimicrobial resistance. So that's why the WHO recommends prudent and responsible use of antibiotics in both humans and in animals as well. So according to the FAO, chickens are the most farmed animal in the world. That's not overly surprising, but it is quite a shocking number. Their estimate's over 33 billion chickens per year for food. And of course, that's far more than the number of humans. And we also don't turn over every year, so quite a large number. Throughout this presentation, I'll be giving some Canadian statistics and some American statistics, depending. But one interesting thing in Canada fairly recently is that category 1 and 2 antibiotics, so these are the ones that are most important to human health, category 1 being the most important and 2 being slightly below that. So these were actually removed in Canada in 2014 and in 2018, respectively, for the preventative use on Canadian poultry farms. So you can no longer apply these without birds being sick. And if they are sick, you have to basically have a veterinarian determine whether they would be reasonable to use or not. So that's interesting. No more just sort of indiscriminate usage of those. And of course, the reason for that is the concern associated with AMR being seen in human medicine. And we want to conserve these very, very good antibiotics for human uses when we get sick. So that was a nice step forward. They're actually looking at potentially removing category 3 antibiotics in Canada as well for prophylactic usage. COVID actually disrupted that process, so they're taking longer to make their decision on that than they would have. And then category 4, they're not going to bother removing because those are the lowest importance antibiotic, and they're actually not used in humans. So there's not so much of a motivation to get rid of category 4. Okay, so that's sort of the current landscape. So as you can see here, salmonella, not surprisingly, highly associated with farm animals and their products, as well as fruits, vegetables, and nuts, but the vast majority you're seeing sort of between chicken and eggs. So almost half just between chicken and eggs, and then you see sort of a less so in other domains. But I'll get a little more into sort of these different serotypes of salmonella, so these subgroups of salmonella, which actually are more highly associated with each different commodity, and that's depending on the factors associated with that commodity, how they're generated, how they're processed, and then how well that type of salmonella is actually suited to it. But this just gives you a nice sort of overview of how prominent it is, especially with poultry, which is what I'm interested in. So of course, aside from antibiotics, that's one that we're very worried about because of the impacts that it has on human health. But there are many other control strategies in place as well, which you would know. And that's from the context of poultry production. You can see how these are sort of used throughout poultry production. So right at the farm level, you have your sort of agricultural environment. You can avoid things. So preventative things are best, absolutely. So you can avoid overcrowding, for example, which would encourage the exchange of sort of fecal contamination and ingestion of certain pathogens that are associated with the enteric system. You can try and prevent contact with wild animals, which would be potentially contaminating your animals. You can also have measures for people to wash in and wash out as they're entering a barn, so as not to bring in any contaminants. Then once you get to the manufacturing side, of course, we have things like chlorinated wash water, heat, drying, acid programs where you can look at critical control points, environmental monitoring to make sure nothing funny is happening, and sterile packaging. But there's limitations to these things, especially when we think of chemicals like chlorine. They're very important, very useful, but there's a limit to the concentrations we can use, which means that there's the potential for organisms to still be able to persist in spite of those treatments. And then, of course, on the back end, when we talk about distribution, retail, or even the home environments, refrigeration is really the key to ensure that any level of bacteria that you do have isn't able to basically grow and multiply on the food product, and just keeping those levels hopefully very, very low, hopefully at a safe concentration, because of course there is sort of a threshold, a pathogenic dosage, which if you eat a certain number of cells with a different organism, then you're more likely to get sick. So if you can keep those numbers very low, it can definitely reduce the likelihood of illness. And then, of course, hugely important is the cooking at home, but that's up to the individuals in their homes to do it properly, to not cross-contaminate. And then you also have situations in restaurants and things where you have large quantities of food, a lot of commotion, and the opportunity for cross-contamination can still occur. And of course, it can occur at any point throughout this chain. So this is something I wanted to draw your attention to. How prominent is salmonella actually in the animals themselves? And then once it passes through processing, how prominent is the salmonella at the end of the production cycle? So once it gets to you. So if you look at the bottom of this figure, I have British Columbia highlighted in a box there. This is relevant to me because I'm out at UBC. But here, and similarly across Canada, although the values fluctuate a little bit, you can see at the farm level that 2 out of 3 of the birds that were tested, so about 67%, they had viable salmonella within them. So salmonella is interesting. It's an interesting problem with chickens because it actually, the different types or serotypes that are associated with them today, they actually don't make the birds sick generally. Very, very infrequently do the birds get sick from this, but it makes us very sick. So that's interesting right away for just the ability of the salmonella to be present in the animals without them showing visible symptoms. It's more of a symbiote for them. And then you can see as you move up the table, those numbers decrease. But by the time it's on, let's say, frozen raw breaded chicken products, there's about 37% which have viable salmonella that could make you sick on it. And then the chicken breast, it's about a quarter approximately. So that's pretty large when you consider how much chicken is consumed. That's a lot of viable salmonella associated with ready, like not ready to eat, but foods that you're going to consume. So it is, despite all of these different things that I mentioned, which are there to reduce the amount of salmonella, we're still seeing quite a high proportion present there. And this is sort of interesting as well. This is just a look at some of the strains that have sort of been escalating recently. So again, this word serotypes that I keep using, the types, the sort of subtypes of salmonella. And you can see, so I put in the box there between 2014 and 2018. So remember in Canada, and these are Canadian figures again, we removed the category 1 and then the category 2 antibiotics. And all of a sudden, we're seeing a very large increase in Salmonella Kentucky, which I'll talk about more. Enteritis is another type that you've definitely, I'm sure you've heard of, as that's the most common. And then on the right, you see with turkey, there's Salmonella reading, it's called, and that all of a sudden has gone up quite a bit. I will say I'm not insinuating that the removal of antibiotics has been causative for the increase of these certain strains, but it is interesting, and it'll be worth continuing to monitor these to see how the serotype prevalence changes and maybe what genetic factors are leading to that change as the system might be trying to adapt as we move forward. So very simply put, I think I've made it clear enough that salmonella is a problem, especially when we're talking about with chicken. So we want to use bacteriophages as another hurdle, which we can implement to target that salmonella within poultry production. And you saw along the chain, there's all these different spots where contamination can occur, where you could theoretically implement another antimicrobial and potentially get some benefit. So part of that is also collaboration with these different industrial stakeholders and sort of defining what their limitations are and where it makes the most sense to target. So, but just to give you sort of a refresher on bacteriophages in case you're less familiar with them, these are bacterial viruses. And you can see in the image on this slide that there's all of these little green things sticking off. Obviously, this is artificially colored. This is an electron image, but they're much smaller than the bacterial cell, and they bind on, and they can actually inject their DNA and attack it as a virus that only targets that bacteria. So they are actually highly specific to their bacterial targets. They have sort of a host that they prefer, and they're not able to cause an infection to replicate themselves in hosts that they don't recognize or that aren't compatible. Similarly to how some viruses can, let's say, infect humans, but not another host. It's sort of the same thing, but with bacteria. And then, of course, they replicate within that bacteria that they infect, make more of themselves, and then spread and kill more of the bacteria around. And they are actually the most abundant replicating entity on Earth. So there's an estimated 10 to the 31 phages, which is an astronomical number. And of course, that's just a guess. So here's sort of a closer look. You can see that they have that icosahedral head that contains their genetic material. It can be DNA, it can contain RNA, it can be single-stranded or double-stranded. So there is some variability, but generally there's that structure. There may be a tail if they're a tailed phage, which you can see in this particular image it is. And it's basically able to recognize the cell with the base plate and then inject that DNA or RNA and sort of hijack the bacterial cell in that manner. So I'll show you sort of what that actually looks like here on this slide. So the manner of replication that a phage might use can be lytic, which you can see on the one side, and then lysogenic, which is on the other. So we're interested in lytic bacteriophage. So these would be virulent bacteriophage. And basically all they do, they don't have any genes that allow them to sit dormant within a cell. They just basically bind, inject their genetic material, encourage the cell to produce many phage components, self-assemble, burst out of the cell. Very simple replication strategy. That's what we're interested because it makes the most sense as an antimicrobial. And you can see how it's, since it's causing the bursting of the cells, that this would make sense as an antimicrobial as we want to kill those cells, right? So in this case, we would be doing this with Salmonella. What we don't want to use is lysogenic bacteriophages. So these actually have extra genes that allow them to incorporate themselves into the genome of the bacteria. And then they can sit dormant. And then as that cell replicates, the phage is replicated with it. And then upon some stressor, they might then revert to a lytic phase and then make a bunch of themselves. But we're not interested in that whole dormant period because that's not actively killing the cells during that time. And we also don't want to have any integration to have any sort of funny gene swapping occurring. So that's not what we're looking at. And in our research, we've actually genetically screened all of the bacteriophages that we use to ensure that they're lytic because the lysogenic genes associated with bacteriophage are known. So we don't use those. So as a food antimicrobial, bacteriophages offer several desirable attributes, you could say. So they're green technology, that is to say they're sort of clean label. They can be considered organic because we're just getting them from the environment. We're not genetically modifying them, although you can. We're not. They're kosher, halal, whatever. They're quite clean label. So that's good. And people appreciate that in industry as they like to be able to, and they're not labeled as an antibiotic. So of course, you've seen things too, marketing, they're saying raised without antibiotics, things like that. So they are very cognizant in industry of these sorts of labels, which is why it's important to know that. They do kill only the specific bacteria you're after. This can be beneficial in ways as well when we're talking about the microbiome. As we're all aware, there are many different microbes associated with animals and the foods we eat, and then also within ourselves that can be important to health. So unlike broad spectrum antibiotics or harsh chemicals or things like this, we're not wiping everything out. We're just selectively targeting the thing we want to kill, in our case, Salmonella. They are safe for human consumption. We're covered in bacteriophages right now, inside and out. Again, this is also assuming that we're talking about phages that have been selected to be specific to pathogens. So they're not interfering with other things, and they're just proteinaceous. So it's not like the protein that composes them is necessarily causing issues, especially when ingested. They're self-replicating and self-limiting. I described that before, and they're ubiquitous. So you can find them everywhere, and it's just a matter of screening them to find the ones that you want. So then the question becomes, this all sounds very good. If they're so great, why aren't we already using them? Well, there's sort of a complex historical reason for why we're not using them right now. So these are the gentlemen who are credited with originally discovering bacteriophages. So they were discovered independently by Frederick Twort, which is on the top, in 1915, and then also Felix Durrell in 1917. He's in the bottom, and they looked very highly promising. Basically, they saw that when they were looking at bacterial colonies, all of a sudden they would die, and you'd see sort of these plaques emerging. So they'd look clear instead of turbid, which we're used to seeing when we're looking at bacteria. So they thought this was very interesting, and Felix Durrell actually said there must be some sort of virus that's killing the bacteria. So very promising. Felix Durrell actually used this in humans, looking at dysentery with a lot of success. So he found this to be the sort of future, and it was applied in soldiers in France as well. However, bringing us to the reason why they're not used right now, like antibiotics are very largely, is because of Alexander Fleming, who discovered penicillin in 1928. So penicillin, first antibiotic of great use, much broader spectrum than bacteriophages. So you could just apply it and all the things that you wanted to die seem to die when we're talking about microbes. It seemed to be very safe and all of these things. So that sort of was the end of bacteriophage. These things that seem to be very fickle, they seem to only target very specific things, etc. And they're also bacteriophages, since they're proteinaceous and not a chemical. They are actually more labile than an antibiotic is, which means they can degrade under certain circumstances as well, like in stomach acid or things like this. So why are we using phages now? Well, as we know, antimicrobial resistance, huge issue. We're seeing a great decline in the efficacy. We're commonly having to revert back to using sort of these antibiotics of last resort in human medicine. So now we need something to pick up the slack so we can apply antibiotics less and basically conserve antibiotics for human use. That's not to say that you can't use bacteriophages in human medicine as well, but they are currently reducing antibiotics in agricultural settings massively, as I mentioned, with those new policies. And so something needs to sort of take that place. So that's why phage makes sense now. So back to Salmonella, which we're interested in. So it infects humans and animals. It colonizes the gut and it's shed through the feces. So it's fecal contamination in the birds, which actually leads to contamination of the products themselves during slaughter and processing. And that's how it's transmitted. And just to give you some statistics to how bad Salmonella is, how much it impacts society, I've actually included some American figures here, so it's more relevant to you. So it's the leading cause of foodborne illness in the US. It's, according to the CDC, there's about 23% is attributed to poultry consumption. So not surprising, since we saw that poultry commonly harbor this. About 1.35 million infections per year just in the US, and about 26,500 hospitalizations per year. Of course, when it becomes severe enough that people have to actually check themselves in. And then according to the USDA, it's the leading cause of sort of financial detriment at about $4.1 billion per year. And that has to do with, hours of work lost, and then also mortality and then expenses in the hospital, all of these different factors combined. But which is quite shocking for an annual basis for one pathogen, quite a large impact. So a Salmonella infection, known as we all don't want. So fever, chills, diarrhea, abdominal cramps, headache, nausea, vomiting, terrible. And then of course, we're often contaminated through contaminated food or water. And then symptoms last for about 6 to 72 hours, depending on how severe it is, on what strain you actually ingested. And then this is, of course, much worse if you are immunocompromised. So whether you're very young, very old, or you have an autoimmune disease, if you're receiving cancer treatments or things like this, you're at a much higher risk than someone else. So, but in healthy people, so your immune system's intact and the dose that you ingested was not unbelievably high, salmonellosis often clears up without treatment. And the key thing is to stay hydrated. So, okay. So it's important to differentiate when we look at Salmonella, the different subgroups within Salmonella itself, which is the genus. So within the genus, we have the species Bongoria and Enterica. We're interested in Enterica as Bongoria is typically associated with sort of other animals like reptiles and things like that. whereas enteric is associated with animals that we tend to eat. So within that, there are these serotypes. So there's over 2,600 different serotypes within enterica, but then you just see this handful of serotypes which cause sort of all the problems. And that's just due to their genetic adaptations, which allows them to persist and potentially cause other issues. So enteritis and typhimurium are two kinds of serotypes which are very common, but there's some others as well that I'll be mentioning. I had already mentioned reading in Kentucky before, but there is a subset that are sort of more important. So these are the sort of important factors that we can see differentiate between serotypes. So you can have these all relate to sort of the disease outcome within a human. So you can have increased disease severity, as you can see at the top of this slide, with certain serovars, which might be more or less associated with different food commodities, or you can have less severe serovars, and that's because they don't have the virulence factors within them, the genetic virulence factors, which cause certain symptoms in humans. So interestingly, at the top here, you can see that Salmonella Kentucky is actually associated with a low level of human virulence. So this holds true in North America currently, but some work I've been doing, and also I've been speaking to some other labs as well, what's being seen is there's a very high level of genetic variability in Salmonella Kentucky compared to other types of Salmonella. So this high level of genetic variability might give some indication that maybe they're more competent or more able to take up genes from their environment, which is quite alarming because they've become very, very prominent within the food domains, especially with chickens. So that's interesting. So they aren't highly virulent right now, but they contain a lot of genes that are a little bit scary, especially in antimicrobial resistance, which I'll show you shortly. And they seem to be able to pick up other genes, which could mean they could get virulence factors later. Now in Europe, all of a sudden recently, they're seeing certain types of Salmonella Kentucky becoming very virulent in humans. And so I think this is alarming, and I think it's something we need to pay attention to before it gets to a point where we're just responding in a panic because it's so prevalent. Of course, there's other things that you can see on this image that are associated with disease outcome. So I had mentioned that the host health status is important, and then also the strain fitness. So what genes are present here? Is the cell damaged? Yes or no? So I think this all makes sense. So this is a figure from the library of Salmonella isolates that we have in our lab. So we genetically sequenced these and then looked for antimicrobial resistance genes. And you can see along the top of this figure, there's different serotypes of Salmonella. So you can see on the left, we have typhimurium and enteritis. These are highly prevalent in poultry production. And then underneath that, you can see that it's all green. And to the left, on the left-hand column, you can see that there's all these different antibiotics that we tested, and they were found to be susceptible to all of these major antibiotics. However, as we move right along this figure through the different serotypes, we can see these orange boxes starting to pop up. And what that indicates is resistance to an antibiotic that was tested. And so there's a lot of these different genetic variants of Salmonella Kentucky, which I've highlighted in purple. And you can see that a lot of these different variants have high levels of antibiotic resistance, which is very alarming, as I said. Now, in our library of the typhimurium and the enteritis isolates, they're all like almost identical to each other genetically, very, very, very similar. But the Kentucky, they're all over the place. They're picking up genes and plasmids, and it's very alarming. Now, you'll see reading as well, sort of squished between the Kentuckys in one of the columns, and it actually has a lot of resistance as well, this particular isolate that we had. And this is one that's exploding in turkey population, so that's also worth watching as well. So quite interesting and a little bit alarming because we're talking about the ability of antimicrobial resistance genes to be pulled through from the farm all the way to humans, and then and so on. So you can think about it basically like this. So you can see, I'm not sure if you can see my mouse or not, but down at the farm level, you have these genes that allow sort of environmental prevalence. It's getting into the bird, it's lasting in the bird. So then you have these animal specific factors that allow it to basically persist there. Then as you move through the system, they must have some level of sanitation resistance. I mentioned some of the hurdles that they have to get through in the current system. And then they have to have genes that allow them to persist in storage. And then they have to be able to infect humans. And then at that point, do they have antimicrobial resistance genes? Do they have a lot of virulence genes? And those are the ones that we should be really worried about, because those are the ones that we need to theoretically use antibiotics in the hospital to help people who are either immunocompromised or losing the battle against salmonellosis. And that's when we no longer have any options. When you look back at Kentucky, which I showed you, which is resistant to many very important human antibiotics. So the QR code here actually would take you to a paper that I recently published, which goes into much greater depth about this, which you can feel free to look at, or of course you can reach out to me and I can connect you to that as well. So when we look at online databases, we can see that basically this is a breakdown of something I was already saying. The larger the bubble, the more isolates present in the database. But what I really want you to see is that Typhimurium, largely dark blue, as is Enteritidis. So they have been isolated in humans, whereas they're smaller in poultry. But in Kentucky, we're seeing sort of this explosion of Kentucky lately. And again, I think I've sort of told you why I'm worried about that one as is, but it's just another way of looking at it. And then you can see some of these other isolates and how they break down between being found typically in birds versus in humans. So to actually generate a bacteriophage cocktail that could be useful against the isolates that I've been showing you, we started out with our library of salmonella isolates, which were taken directly from broiler chickens. And then we also had a library of bacteriophages, which were isolated from the just environments. Remember, phages are everywhere. And then we wanted to look to see what the smallest number of bacteriophages could be used to target the most number of these relevant salmonella isolates, or serotypes, I should say. Can we make a cocktail of this, and how effective is it? So that's really what the goal was initially. So you could do something called spot testing, where you just have a plate with your bacteria, and then you just put different bacteriophages on it, and you see which ones are able to kill it. And this plate that's shown here, it probably, if you've done a MIC test, or you've ever done like antibiotic disc diffusion assays, you can see that zone of inhibition that's happening. And that's just, it's what we call a plaque. It's areas where there's been death of these bacterial cells, and that would give you an indication that that bacteriophage that you've applied is able to kill that particular serotype, right? So what you can do is then put together a heat map. Like this heat map shown here, it just gives you a very zoomed out look at the comparison of our different libraries, the phage versus the salmonella. And the darker the green, the more effective that bacteriophage is at killing that particular salmonella. So then you can just go from a full library and you can zoom in on the phages that seem to be the most promising. Then you can do something similar. It's called efficiency of plating. This figure is a little bit maybe confusing to look at. It's just a scoring system. And basically all I did was dilutions of these bacteriophages to see sort of how efficiently they're able to kill the salmonella that we're most interested in. And then it's just a more time-consuming way to zoom in even further on the phages that you're interested in. So that's all you really need to take away from that. And then, so these are the serotypes of salmonella that we were most interested in for British Columbia. Of course, the serotypes can vary depending on the geographic location, but most of these are quite, no matter where you go in sort of the Western world, you'll see the same sort of serotypes, generally, some variation. And then these are the phages that sort of looked most promising. And then I was just able to come up with a combination of 3 of the phages from our original library, which when added to any one of these top salmonella serotypes, basically you see a huge reduction in vitro. So what I will say about this is that in your initial, let's say 9 hours, you've basically eliminated the salmonella. But you do see this rebound effect here, which is interesting. So the thing with this particular test is you're giving the conditions for the salmonella are absolutely perfect. It's 25 degrees Celsius, which again, it's lower than it prefers body temperature, let's say, but it's a very nice growth temperature for the bacteria. They're in high nutrient media. The bacteriophage was only added once. There's no bacterial competition. So it's not a perfect parallel to if you were to use it in industry, but it does give a great idea that, yes, this bacteriophage cocktail that contains only 3 bacteriophages is capable of knocking out all of these very important salmonella associated with poultry. So that's what you should take away from that slide. There's other ways that we've looked at these phages as well. I told you we looked at them genetically. This is just looking how long does it take for them to actually infect a cell, and then how many bacteriophages are produced from each cell. And you could feel free to look at the paper if you want more information on that. And then these are transmission electron microscopy images of the different phages that were my favorites. And then the ones in boxes represent those that were actually used in the cocktail. So that was really fun taking those images. And this is the paper if you're interested, which it goes into more depth about generating that bacteriophage cocktail and the considerations there. But I'll just move past that for now. So then we have an understanding of the salmonella that's important to industry. We have an understanding of which bacteriophages can be used to actually target these salmonella and how effective they are, at least in the lab. But it's also important to understand when you're talking about from a One Health perspective, what is it that your customer segment, what's the value proposition to them? What pains do they have? And what do they stand to gain depending on how you implement the theoretical treatment? So in order to have a solution metaphorically taken off the shelf and implemented, you have to understand your market, basically. So because they have way more than just the one problem that they're trying to juggle, salmonella is on their radar, and we saw how detrimental salmonella is to the individuals within society. But they have many, many, many other problems that they have to deal with. So coming up with sort of this critical control point or a subset list of different sort of high impact application zones is best case scenario. That's what you want to do to actually have an impact within the domain of interest. So through my PhD, which I did in Dr. Xian Wang's lab, we spoke to many different stakeholders who have interest at different levels. So different farmers, policymakers, inspectors, veterinarians, researchers, industry, and all of them sort of had their thoughts, their 2 cents about how to go about things. But then by concatenating all of that different, all of those different conversations, we were able to find some ideas that we thought would maybe increase the impact of application of our particular cocktail. So what seemed to be one of the key features of the salmonella in poultry problem is the abattoir. So you can see in this image, it looks like a bottleneck because it really is in the modern production setting. It is a bottleneck between many of these different farms, which each can have their own salmonella issue. They can have different concentrations. They can have different serotypes. Again, being unrecognized because the birds are not showing visible signs of holding the salmonella. Then you have them go sent into a single abattoir, which is a slaughterhouse essentially. And while they're being processed, you're having all of these different diverse salmonella basically aerosolized onto the meat, which is why initially earlier in the presentation, you saw those high percentage of contamination of viable salmonella. It's happening here because it has to go from their digestive system to the meat. And then what you're having is then a distribution from that abattoir to many other stores. And you can see these are some values that I've shown for different provinces in Canada. On average, there's about 15.5 producers shipping to a single abattoir, but province to province, it varies. And for example, in Quebec, you have almost 55 producers shipping to a single abattoir on average in that province, which is quite alarming. You can think of all the diversity of microbes being put into that one location and spread around. So it's a complex problem in that way. So our idea was, for the sake of this project, let's look at the front and the back of the abattoir. What do we have? We have live mature broiler chickens going in, and we have meat products coming out. Could we treat these as our critical control points and have a very large impact on dropping that salmonella before or after the abattoir, basically before it gets to human consumption? So this experiment was one of those tests that made me feel like a crazy person. just because I was like measuring and weighing all these little pieces of chicken and then treating them with salmonella and bacteriophage. But we decided to use chicken breast initially because it's just a nice, consistent food matrix. We'd just be able to see proof of concept. Remember earlier when I was doing the in vitro test, it was in a broth, which is perfect for the bacteria. There's no competition. This, on the other hand, was purchased same day from a store. It's exactly what you would get if you went to the grocery store. So it felt representative for an early sort of meat test. And then we used a cocktail of different salmonella, which was the most prevalent. So they are the most prevalent basically everywhere in the Western world right now, which would be Typhimurium, enteritidis, and Kentucky. And then we applied that to the meat as a challenge, allowed it to adhere. And then we applied our bacteriophage after as a mist. And then what you can see here, just from this simple figure, is that the orange line, which is the chicken breast, which was challenged and then not given a phage treatment, it started out with a decent level of salmonella, colony forming units per milliliter. And it continued to grow because we left it at 10 degrees Celsius, which is refrigerated, but not below the growth temperature of salmonella. This is also a temperature that can be commonly found for chicken as it's moving, say, from one location to another. Although it's refrigerated, they find the temperature can go up to about 10 different segments, especially when you're taking it home. But then when in that blue line, you can see it's very subdued, the amount of salmonella. And it stayed subdued for 24 hours. It did go up a little bit after 5 days, which is arguably a little bit extreme at 10 degrees Celsius, but it had a great effect. And there's a paper that's going to be released on this very shortly, actually, if you're interested. And there's more information about different treatment features and things like that. And since then, we've actually optimized slightly further. And we found that we can actually flatten this line when treaters just by making a few tweaks to our formulation. So very promising. Again, over 99.9% reduction. And you've got to imagine if it comes down to, would you rather ingest 100,000 cells or 10,000 cells versus 1 cell or 10 cells, right? So it can be the difference between getting sick or not, this level of reduction, even if you're not completely eliminating salmonella. Although in some circumstances, we did completely eliminate it. So that's at the back end of the abattoir, which is the bottleneck. But at the front end, we thought, okay, what is the least amount of treatments that you could do just to, reduce costs? Because that was something that kept coming up when we were talking to stakeholders. Cost is always a huge issue. So what is the least amount you could do before those birds go into the abattoir to have a meaningful impact on the salmonella load? So we thought, okay, mature broiler chickens, they could receive 1 dose or 2 doses sort of in a window of time before they're shipped off, and then that could knock things down right before they go into the abattoir. So the thing with bacteriophages, though, is they have a proteinaceous capsid, which means that they can be destroyed in the upper gastrointestinal tract of a mature broiler chicken, which it's very acidic, and then there's some grinding action. So we decided to put them in these enteric capsules. We put the phage, we suspended it in a microemulsion, and then we just packed them in a raisin and then allowed them to eat it. And the idea was, yes, this is a very simple way to just deliver sort of a quick shot of phage in a mature bird, which is fairly large. And again, if it just drank it, it would be on the beak, but it wouldn't target anything lower in the digestive system. So this was our sort of workaround for minimal dosing in a mature bird. Different groups that we had set up for this experiment are shown here. So we had a group that was just received the salmonella challenge, no phage, and then we have a group where they got their phage 24 hours before the salmonella challenge. Group 3 is the same day as the salmonella challenge, and group 4 is the day after the salmonella challenge. And we just wanted to see how this affected sort of the dynamics of what levels of salmonella remained in those mature broiler chickens if you staggered the phage treatment. And this would give us an idea of how long do these phage persist in the system, how effective are they, the further away you get from the salmonella challenge. And so I'll show you sort of what that looked like. But first, here's a few pictures of the young chicks early on in the study. Of course, we had to grow them up a little bit before we could test on mature birds, but they're very cute when they're small. And that's one of the vet techs as well, holding a bird, sort of the teenager bird on the right there. And then within 5 weeks, they actually get very, very large, and they're ready for, this is usually the size that they would be shipped off on. So 5 weeks, I was quite surprised actually when I first learned that that's all it takes to grow a modern broiler chicken. So this is sort of the little setup we had for that experiment. So what did we see? So these figures depict the results for the how much viable salmonella, I should say, was present, and we were able to do this through cloacal swabbing and then actual presence testing on XLD agar. So this is from their feces that we were able to see this. And so for the early one, which is on the left, so this is the group where you can see they got a bacteriophage cocktail a day before the salmonella challenge. And what you see is a slight reduction, but it actually wasn't significant. And then we actually did do a second treatment, and we saw potentially another reduction, but again, not significant, so I can't speculate too much. So you can have the salmonella getting there too early, not persisting within the system, and then not causing maybe a meaningful reduction. So it's good to know. For the late phage group, so group 4, again, this one, they instead received the bacteriophage after being exposed to a high dose of salmonella cocktail. And we saw maybe what you would say is maybe a more meaningful reduction here, and it sort of stayed lower, the amount of salmonella compared to the control that did not get exposed to phage treatment. However, this was also not significant, which might show some indication that yes, we're causing a reduction, but we're not, the dose might not have been high enough, or maybe one dose isn't enough. Fair enough. This is the on time. So this is group 3. So the salmonella and the first bacteriophage cocktail capsule were given to the birds on the same day. So this would be sort of the best chance for the phage to be present at a high concentration to actually eliminate a lot of the salmonella within the system. And we did actually see a significant reduction in this group, which means that presence of that high concentration at the time of infection seems to be important. And that could be just an issue of the phage being able to physically find and interact with enough salmonella to pick that up on the back end and see a reduction. So what does this say to me? This says our dose may not have been high enough. We were using, due to the constraints of using the capsule system that we had, we could only use about 10 to the 8 PFU bacteriophage. So maybe using 10 to the 9 or 10 to the 10, or multiple capsules would be better. But this is still really nice proof of concept. And it did show that delivering it down to the lower levels of the GIT where salmonella is harbored in birds, you could actually target it. So that was great to see. And it would just be a matter then of sort of refining our tack and basically improving the impact that we saw. And then also timing things so that that lowest level was occurring when the birds were being sent to the abattoir. We did see sort of this rebounding effect throughout these trials where salmonella would go down and then rebound, and if we applied again, it would go down again. And that's worth knowing because timing your treatment would also be important for the industry, again, to minimize the cost, minimize the number of trials, minimize the stress on the birds. So yes, I would call it a success. So this bacteriophage, as I had mentioned before, extremely specific. They just sort of see salmonella. They can't see anything else unless maybe it's very closely related. But what this means is the microbiome of the birds, which we then looked at, stayed intact, which is excellent. That's not something you could say with a harsh chemical or a very strong broad spectrum antibiotic. This shows the growth rates of the birds and across the different treatment groups. No significant difference. They all grew the same. They all looked the same. Veterinarians monitored their health status. They looked great. So that was excellent to see as well. And then to close the study, we actually took sort of a representative sample from the upper, mid, and lower gastrointestinal tract of the birds that were involved in the study. So for the upper, we did the duodenum, for the mid, the jejunum, and for the bottom, the ceca. And we wanted to see if there was any changes in the microbiome throughout the digestive tract to see if maybe the phage were causing any issues that were noticeable or drastic. We didn't expect to see anything because, again, phage are specific. The birds looked very healthy. But just to quickly look at that, the beta diversity, what you're seeing for each of these. So you're seeing basically overlapping. On the left, you have the upper GIT, which is duodenum. In the middle, you have the jejunum, and on the right, you have the cecum. And the different colors represent the different groups. But what you're seeing is they're just overlaying each other in these different plots, which gives an indication that the beta diversity is not significantly different. And in fact, the bacteriophage are not causing drastic microbiome shifts within the birds at any of these segments of their gastrointestinal tract. So this was great to see. It's what we thought would happen, and it is what happened. This is another look just through a different beta diversity metric. It's showing the same thing, really, so I'll skip past it. And then when we looked at the presence of the different microbes within this, the gastrointestinal tract of the birds, so we looked at this through 16S, our RNA sequencing. What you can see on the left side here, these, so you can see there's a lot of gray and there's a lot of blue between these different treatment groups. So these are the main organisms that were found within the cecum of the different birds. And they're largely unchanged. They're essentially the same. Slight variations you can't really say too much about due to the nature of 16S RNA sequencing, but they're largely intact, which goes to show that these bacteriophage are not decimating important, highly prominent microbes within the GIT, specifically within the ceca. And then this is just on the right here, figure B. This is just a zoomed in of these top less prominent organisms found within their cecum. And what we can see is that in the group that received no bacteriophage, they have this large green bar present here, which is the number of Enterobacteriales, which Salmonella does fall within. We were limited in the amount of resolution we could achieve with this method, but it does give some indication that, again, maybe we're just seeing a reduction in these types of organisms, which the phage would be able to actually recognize. But other than that, there was no real changes. So that was good to see, and it was pretty much what we thought we might see. So in conclusion, despite current methods, obviously foodborne illness is still a threat. Salmonella is one of sort of the big 5 that we see in the food system frequently. Phage biocontrol is an additional tool in a multi-hurdle approach to prevent foodborne pathogens from reaching consumers. So we're not saying get rid of chlorine, get rid of temperature controls and all these things, keep the birds healthy. This is one more hurdle we could throw in to hopefully bring those numbers down again and increase the resulting food safety. And then phages could allow food processors to preserve the beneficial food microbes while strategically targeting the pathogens. So that's great. When we're talking about in live animals, keeping their microbiome intact. When we're talking about on chicken food surfaces and things like this, bacteriophage have also not been found to cause organoleptic changes, which is excellent. So you wouldn't even know that it was on there as a consumer. So with that, yeah, thank you very much for having me. Again, you can scan this if you want to add me on LinkedIn or ask more questions later. But other than that, I welcome your questions.
Thank you, Thomas. It's a great presentation. I think it's beautifully made slides, very impressive work. I'll let the audience have questions first, and I have a couple of questions for you as well. Maybe I can get started with, so the first question I have is on your slide number 8, you show the prevalence of Salmonella in different provinces, is that called, in Canada?
Yeah.
Yeah, in noticeable Ontario, it seems they have much lower problems of Salmonella. It's like way lower than the other places. Yeah,
it varies from year to year and also from province to province. But British Columbia actually is known as being one of the, I think it is actually currently the worst for Salmonella. It could be, there's theories as to why that is. The practices used across the provinces are largely the same, or generally the same. But the climate in British Columbia is actually much warmer than the rest of the country. It's much wetter.
Right.
It's possible that this is lending to the ability of like high proportions at the farm level, which then trickles through. But that's speculation. I've talked to veterinarians about that too. That's kind of what they think.
Yeah, that's what I guessed.
Mhm.
On slide number 27, you showed the treatment on the chicken breast. I suppose you spray on the surface of your phage solution, right?
Oh, this one was this was actually in a 96 well plate.
Okay. One of the picture you show, you have a lot of chicken breast on the table. Yes.
Yes, this one.
Yes.
So this is directly on chicken breast. Yes.
Okay. So it looks like it's gonna take about an hour up to 3 hours to see some inactivations of the salmonella. Is that in general the
So it actually happens very quickly. You see a reduction in the salmonella almost immediately. I would say within an hour.
okay?
And the thing is, so this was done at a temperature where the metabolism of the salmonella would be active, but not fast, like it'd be replicating slowly. But if you even halted the metabolism of the salmonella, so say you're doing it at 4 degrees Celsius, what you get is a bunch of bacteriophages punching holes on the outside of the cells. It causes something called lysis from without. So even when you halt that, you can get immediate death of the cells just by punching holes. But it's quite quick. Yeah.
Okay. Yeah. Okay.
And at the concentrations we use, it's basically instant because you're just getting all those holes punched in the cells. The phage actually don't even have to replicate necessarily to kill the cells.
Right. One last question from me. I love the idea of you making a capsule for the phages and feed the chicken. And I wonder in the practical use, what's the cost of making the capsules?
Yeah, so you're right, it does increase the cost. I would actually not recommend doing the capsules as we did them if you were going to use them practically. This was more just, we were seeing, could we use a low dose of phage and target salmonella and cecum? It was more about reducing the amount of salmonella that we actually, or sorry, reducing the amount of phage that we actually used. So yeah, the cost would go up with the type of encapsulation you decided to use. This was just a quick and easy one for us to do to prove our point. But ideally, you'd want to encapsulate to protect from the upper GIT in the most affordable way possible. To create the phage themselves as a liquid, which you can then work with, is extremely cheap, unbelievably cheap, because it's just the cost of growing bacteria, really. That's all it is. So again, you also even have to think about things. So you're right, the cost of those types of pills we did might not be appropriate. But on top of that, you have to even think about distribution. So these capsules were they were not big, but they were big enough that the birds wouldn't necessarily just go around and eat them. So you would even want to shrink them down if you were to do something that was to be applied within the actual market.
Okay.
Yeah.
Wonderful idea, interesting work. TJ, you have questions?
Yes, this is a wonderful presentation. I'm with the Food and Drug Administration. I'm actually a research chemical engineer, so I don't have a lot of background on phage. Will they multiply by themselves, or they just stay, I don't know, like active when you apply to the food?
Yeah, that's a great question. So they will actually replicate within the cells that they target.
Okay.
There is some nuance to the dynamic. So for example, if you have many phages finding a low number of bacterial cells, then they'll just kill them without replicating because they'll punch too many holes and cause lysis. If you have sort of more of an even distribution, again, think about the food surface as massive, and these phages are tiny and the bacteria is tiny. So if you get more of an even distribution, they'll replicate and then they'll sort of kill all the neighboring bacteria as well. And then if you have a very low amount of the bacteria and a high amount of the bacteria and a low amount of phages, then again, that will propagate through that population. So it's all about sort of how close they are together and how much phage to bacteria ratio you have.
Okay, so in this figure you are showing now, so in the first day it kill or prevent the growth. But then after that, you can see the growth come back, right? And then if the trajectory seems like it can go just parallel to the untreated control. So what does that mean? What that means all the phage are gone, or is there a resistance? Or I mean, so right. So do you know what? Can you explain those results?
Yeah, absolutely. So the trick is to use I forget which paper it was, but I read in a paper they called it the scorch the earth method. So the idea is that you actually calculate the number of bacteriophage that you need to cover the surface so that the bacteriophage end up close enough together that you can't have basically populations of bacteria growing between bacteriophage. So part of it is the ability of the phage to actually interact with the cells, because you might think initially all the phages that are going to kill the bacteria that come into the contact with the bacteria and kill them, but any bacteria that's not hit can propagate as if nothing's happened. So I think that's part of what we're seeing there. Another part could be resistance, as bacteria are capable of gaining resistance to phage. But what I found interesting with my live bird test was that upon application of a secondary treatment, we saw another reduction. So I don't think it's just resistance, but I think it is more of the physical nature and ensuring you have enough. So for this particular test that I'm showing here, this was not an older, it's slightly older than some of the things I've been doing lately. We hadn't modified the concentration and some other aspects of the formulation. I think that it was physical, not interacting with some of the bacteria. Whereas now when we've increased the concentration slightly, you're not seeing that rebound, which gives me an indication that you're getting sufficient coverage of the food. So using finer mists and the appropriate level of phage is important for not seeing a rebound.
Right. So now if I apply this to, I don't know, produce where the surface is not very smooth, and then how can you reach it by your mist, right? So
exactly.
So, yeah.
Yeah, you have to apply it in a manner that addresses that. So for example, if you had tight ridges or folds or things like this that can be protected from an application, you have to ensure that you have the sort of capacity to penetrate into those folds. Maybe in that circumstance, you'd actually want to use some level of pressure to force things in there. But that's just, off the top of my head. It's very important to have physical interaction between the phage and the cells.
Right. So, well, maybe engineer can come to help with the transport of phages to the cells.
Yeah, let me know. Absolutely.
We can get Greg to model it. Well, anyway, he's not here. But anyway, thank you so much. It's a really nice presentation.
Thank you.
Okay. Any additional questions, please feel free.
Hi, I have a question. I'm Catherine. I'm a PhD student in the nutrition. I'm wondering if, what the practicality is of a phage type, and I don't have a lot of microbiology experience, as I'm sure you'll realize in a second, what the possibilities are for bacteriophages in resistant bacteria that are relevant to humans. So I'm thinking about in hospitals where we have a high proportion of people with resistant C. diff, and whether or not, it might be something similar where like when people come into the hospital, they just sort of prophylactically get some level of bacteriophage for that particular strain to prevent like C. diff infections. Does that seem like something that's theoretically possible?
Oh, absolutely it does, yes. So what's really interesting, what different researchers have found is actually, so the bacteria, when we're talking about in a real system, like a complex system where there's lots of different microbes, there's maybe limited nutrition and whatever, like in a human body, for them to access, the bacteria has to make a choice. Am I going to be resistant to this or this? And so what we're seeing is that the more resistant a bacteria is to antibiotics, the more susceptible it tends to be to bacteriophage implementation. So what we might be able to do is have sort of this 1-2 punch where you can use both, and the bacteria is just not able to be simultaneously resistant to both due to the trade-offs that are required to be resistant to either.
Thanks. That's super interesting.
Yeah, thank you.
Any additional questions? Thomas, you mentioned in the at a late slides, you said the capsule technology allows you to kind of package in 10 to the 8th CFU or PFU of macrophages. Could you explain why that? Why can you not?
Yeah, so absolutely. So at the time, when we did run this test with the bacteriophages that we used, I was only able to propagate the bacteriophages at a certain efficiency. So I had a theoretical limit to the concentration I could achieve through standard means at the time. Additionally, when I suspended it in a microemulsion for reasons that I'll be publishing soon to protect it, based on the size of the capsule that the birds would be able to eat, I had a limit of the volume I could use, and then on the other side, I had a limit of how concentrated I could make that volume. So theoretically, and like since then, I've actually optimized things so you can increase that concentration for sure. I was just a little bit limited at the time, and it seemed like it was sufficient for basically showing, can we use sort of a single, not high dose to have a targeted effect, and then we showed we could.
Yeah, I'm asking because I've seen a lot of probiotic dietary supplements on the packages. They all label like a 10 to the 8th or some 6 billion or something like that, around 10 to the 8th CFU right, for the capsules. So I wonder if it's a packaging limitation for this type of technology or is something else. I'm just curious.
Yeah, so it is. Yeah, it is to a large degree. It is a limitation of the actual packaging, and there's ways that you can increase concentrations, but there's a definite limit. There's absolutely a limit, and so some of the phages we're working with now, we can get up to 10 to the 11, which is amazing, incredibly concentrated. And then furthermore, the trick though also is not using these expensive concentration techniques, because again, we're trying to keep this in the lens of as realistic as we can, and sort of showing what the true impact is, so it can be implemented in a focused and realistic manner. So yeah, it's a great question.
That's right. By the way, I love your gut microbiome work. It's fantastic work.
Thank you.
Okay, I think that will conclude our seminar for today. Really appreciate your time, Thomas, for sharing us with a fantastic research. And if you have additional questions, feel free to follow up with Thomas through email or LinkedIn. And again, thank you for your time, and thank you everybody for joining us today in our research seminar. And wish everyone has a great day.
Thank you very much.
Thank you,
everybody.
Okay.
Thank you.